Hello everyone, this is Ghairatullah Shinwari, and today I'm going to be talking about irreversible cell injury. In my previous video I have talked about reversible cell injury. It's available on our YouTube channel Medix. If you haven't watched that, please go have a look. So, as I have talked in my previous video about reversible cell injury and the causes which can bring in reversible cell injury, I have said and given an example of ischemia or hypoxia, which means low blood flow for an organ or less oxygen in blood, which normally causes cell injury. But it depends on an organ and the severity of stress which kind of cell injury would occur. In reversible and irreversible cell injury, there is two major difference. One, it's the mitochondrial dysfunction, which means even if blood returns to the particular cell which was facing ischemia, mitochondria won't be able to reverse its function, which is ATP generation. So this is the first point. The second point would be damage to the plasma membrane. So as I have drawn here, this is a cell and this is the membrane, plasma membrane, which is normally bilayer phospholipid. Normally the whole plasma membrane is the same thing as this one, but I have just drawn this as an example. This is a lipid, and as well this other one is also a lipid. And when I say bilayer phospholipid, so there is two lipids that combines to each other and makes this whole plasma membrane. So as I mentioned, two major differences between reversible and irreversible cell injury. The mitochondria in irreversible cell injury is not able to return its normal function. It will lose its function. And the second thing will be damage to the plasma membrane, this one. So there is two major differences. The first thing in irreversible cell injury would be high calcium influx to the cell. As we all know, calcium is highly concentrated outside of the cell, but inside the cell the calcium amount is much more less than the outside plus in the blood. So calcium available in the blood and in the interstitium space is much more higher. In irreversible cell injury, calcium will influx from outside of the cell to inside of the cell, and when it goes inside of the cell, it would be a high concentration of calcium. The first thing that the calcium would do, it will go and deposit in mitochondria. And when it deposits in the mitochondria, mitochondria will lose its function completely, and it won't be able to reverse its function. So this is the first thing which is going to occur. Calcium is a very good stimulator for enzymes, intracellular enzymes. There are different enzymes available inside the cell, such as one of them is phospholipase. Phospholipase is available in the cell membrane. If this is the cell membrane, it will be available here. Phospholipase. I won't read the whole thing, but just for an example. And when this is activated through calcium, the first thing that a phospholipase would do, it will destroy the phospholipid which is available in the cell membrane. So now I have a damaged cell membrane which is destroyed by phospholipase. So the result would be I have damaged mitochondria which is not functioning, which is not producing ATP. The second thing, I have a damaged cell membrane which is not functioning anymore. And to regenerate this cell membrane, let's say I need more phospholipids because this is made of bilayer phospholipids. if it's reversible cell injury and the ischemia is no longer available or the hypoxia is no longer available, so my cell will try to generate more phospholipids. It can do that. It has the ability to make phospholipids, but it will require energy, ATP. So if it's reversible cell injury, my cell will try to compensate, which means regenerate these phospholipids, and it will be okay. But in irreversible cell injury, I don't have the mitochondrial function anymore, so I don't have the ATP. And if I don't have the ATP, I'm not being able, my cells are not being able to make these bilayer phospholipid. And this will eventually destroy the cell membrane. And once the cell membrane is damaged, the whole thing just begins, the irreversible cell injury. So there is another enzyme which is called ATPase, this one. And it is responsible to destroy the ATP I have. So I have already dysfunctional mitochondria, which means my cell is not being able to generate ATP, and plus there I have another enzyme which is destroying the ATP that I have. So this results in depleted ATP, which means I have no more ATP for nothing. So I'm already low on ATP. Another enzyme which would be activated is protease. As I have mentioned before, calcium is a very good stimulator for enzyme activation. If it's higher concentration inside the cell, it will, and it has the potential to activate all the enzymes available in the cells. And there are different enzymes available in the cells, but they are not activated at the time, and it won't destroy your cell in normal function. So protease, if this enzyme is activated, this will damage the cytoskeleton. I have drawn the cytoskeleton already here. The cytoskeleton normally, if you look under the microscope, it will look something like this, a normal microscope. But if you see it under the electron microscope, it is made of 3 components, which means microfilament, I have zoomed these 3 structures, which makes my cytoskeleton, the complete cytoskeleton, which is responsible to make the skeletal of the cell. This is the responsibility of the cytoskeleton. But normally, if you zoom that in, it's made of 3 components: microfilament, intermediate filament, microtubule, and intermediate filament. So these 3 components make my cytoskeletal, which is responsible for the skeletal of the cell. But if protease is activated, it will destroy the cytoskeleton. And once the cytoskeleton is damaged, my cell won't have the normal structure anymore. It will lose the structure it has. And it won't be reversible because I don't have the mitochondria to regenerate that cytoskeleton. Another enzyme which would be, actually it's 2 of the enzymes which will be activated, is protease plus endonuclease. And these enzymes are responsible for DNA damage, which is inside the nucleus. It will damage the nucleoproteins available inside the nucleus, and it will damage the DNA. So once the DNA is damaged, the nucleus will follow these 3 steps, which is kinosis. This means that the nucleus will be condensed. It will kind of shrink, and the DNA available inside, it will be divided to big particles, and the second step that it will follow is karyohexis, which means that in kinosis, the DNA of the nucleus was condensed, but here it will be divided to big particles like this. And the last step that it will follow, which is karyolysis. These particles, these big particles, would be lysed, and it will be divided to very small particles. Seeing this DNA material, there would be another protein, which is pro-apoptotic proteins. This will be activated, and once this protein is activated, it will eventually, if I say in simple words, kill this material through apoptosis, which is a procedure which I will explain in my next video. So the last step in irreversible cellular injury would be lysosomal damage. As I have mentioned, in irreversible cell injury, that's the hallmark of irreversible cell injury. There is cell membrane damage in particular, and in general, all the membranes available inside of the cells, which means there are organelles that have cell membrane that have membranes, like lysosome, like mitochondria and etc. These all organelles have membranes. So once the lysosomal damage occurs, which means lysosome is damaged, the membrane of the lysosome will be damaged, and it would be damaged because low oxygen and acidic pH inside of the cell. And once this is damaged, there are about 40 kinds of different enzymes inside lysosome. A few of them which I've mentioned here are hydrolase, phosphatase, glycosidase, lipase, protease, DNase, and etc. So each of this enzyme is responsible for a particular destruction, which means it will destroy a particular material. Like hydrolase, it will lyse anything liquid. Like DNase, it will see DNA and it will destroy it. Lipase, it will destroy lipid. So the result would be cell death, and cell death will occur in different mechanisms, which I will talk about in my next video, and like necrosis or apoptosis. The one way that we can, in exam for example, that which kind of cell death has occurred is our blood serum. For example, if there was a patient with MI and a blood test is done and there was irreversible cell injury to the cardiac cells, in your blood, in the blood of the patient actually, we would see cardiac troponin. Cardiac troponin, which is an enzyme in the cardiac cells specifically. Why would we see cardiac troponin? Because irreversible cell damage has done, and the enzymes which was inside the cardiac cells actually has leaked out, and once it's leaked out, it will be taken by blood. So if it's taken by the blood and you do a blood examination of someone who was just faced MI, you would see cardiac troponin, which is a specific enzyme in cardiac cells. So this was all about irreversible cell injury. In my next video, I will be talking about necrosis and apoptosis, which is a mechanism of cell death. For further medical information and medical videos, please keep watching our YouTube channel. Please like and subscribe. Thank you.